This is a presentation on bacteria and antibiotic resistance for Dr. Irwin in the Biology 1107 course. To begin, we're going to define what bacteria are. Bacteria are classified as single-celled microbes, a member of a large group of unicellular microorganisms that have cell walls but lack organelles and an organized nucleus, including some that can cause disease. There are many different types of bacteria. They are easily grouped into different sections: rod, spherical, spiral, corkscrew, and comma. These single-form bacteria are often grouped together and easily identified. Diplo is obviously in pairs, Staphylo is in groups or clusters, while Strepto is in long chains, such as in Staphylococcus. When discussing bacteria, we often think of the diseases that they cause, such as staph, strep, MRSA, or many other diseases. Many people each year die from diseases caused by bacteria. But bacteria isn't always bad. Many times, bacteria aren't always as harmful as you might think. Before you go running off to the bathroom to wash your hands and scrub those germs off of you, you need to know that there are two separate types of bacteria. There are bad bacteria and good bacteria. Bad bacteria are considered to be pathogens, diseases that can cause illnesses. These pathogens invade your body and cause you to be sick. Many diseases can cause harmful effects. Too often when we take antibiotics, we end up killing the good bacteria that live in our gut. This good bacteria are called probiotics. These are like little warriors that enable you to break down food, fight off other infections, and maintain a healthy environment inside of your body. There are many different ways that we receive these probiotics. People often ingest them without even realizing it. Oftentimes, yogurts can contain probiotics. Other probiotics are picked up during your birth. As you come through the birth canal, you pick up many different bacteria that live inside of your mother. Bacteria reproduce through a process called binary fission. The bacteria doubles its DNA and genetic material, then slowly divides, creating two daughter cells. Bacteria can do this often. Some bacteria can even double its DNA and split in up to 10 minutes. Other DNA take even longer. This is a quick video explaining how quickly bacteria can double. The doubling time for different bacteria varies. This video is a sped up version of a bacteria as it splits and continues to multiply. Multiplying bacteria can be very dangerous. Now we will move on to discussion of antibiotics. Antibiotics, as defined by the U.S. National Library of Medicine, is having the ability to either stop bacteria from reproducing or kill them. The first antibiotic was penicillin, discovered by Alexander Fleming in 1928. While oral antibiotics you take through the mouth and digest in the stomach, these antibiotics are often stronger than the topical ones. Common oral antibiotics are penicillin, cephalosporins, and sulfamides. These different antibiotics work in very different ways. When you take an antibiotic, an oral antibiotic, it dissolves in your gut. There are two different types of antibiotics. They are considered to be bacteriostatic or bactericidal. Bacteriostatic antibiotics choose to stop DNA replication as well as the metabolism or enzyme activity of the bacteria. Bacteriostatic antibiotics also work by preventing protein production inside of the bacteria. Bactericidal antibiotics work by dissolving the cell wall around the bacteria. At this point, our own immune systems will be able to kick in and effectively kill any bacteria left behind by the antibiotics. With the advancements in science and medicine, a person might begin to believe that antibiotics are completely harmless. But if not taken properly or misused, antibiotics can become quite a problem. Antibiotic-resistant bacteria are clever little superbugs. Once you are sick and receive antibiotics for your disease, oftentimes many people start to feel better and quit taking their antibiotics. This can cause a problem. Laying amongst the rest of your bacteria is a superbug, or an antibiotic-resistant bacteria. This antibiotic-resistant bacteria can be left behind after taking your antibiotics or not finishing your antibiotics. This antibiotic-resistant strain may then begin to reproduce and multiply, causing you to become even sicker than you were before. Antibiotic-resistant strains can come about in many different ways. Pharmacists often dispense prescription-only drugs to patients who do not have a prescription. This happens more often in other countries than it does in America, as it is easier to obtain. In a study conducted between 2002 and 2012, researchers found a correlation between the administration of antibiotics and the number of people infected with resistant strains of gonorrhea. This correlation is often scary, because as more antibiotics are given, more antibiotic-resistant strains are produced. This study found that there was nearly a twofold increase in antibiotic-resistant strains of gonorrhea between the years of 2002 and 2012. There are many different causes as to why the antibiotic-resistant strains are beginning to increase. Many scientists now say that overuse and overprescribing antibiotics is what is causing these superbugs to grow in number. Antibiotic resistance is a larger issue, and the CDC and the World Health Organization have considered it to be a crisis. The overuse of broad-spectrum antibiotics has caused many strains in both hospital and community settings to become resistant. Overuse and overprescription of antibiotics is developing in countries like India and China. Another leading cause of antibiotic-resistant strains of bacteria is the fact that many people, once they start to feel better, quit taking their antibiotics. This causes a huge problem. Bacteria are often known for horizontal gene transfer. This refers to the transfer of genes between organisms in a manner other than traditional reproduction. When people quit taking their antibiotics, they often leave bacteria behind. Horizontal gene transfer is the primary reason for bacterial antibiotic resistance, and plays an important role in the evolution of bacteria. So in conclusion, in order for us to stop more superbugs from being created, we need to make sure to learn more about our antibiotics and how to properly use them. Weak infection prevention and control practices lead to the increased transmission of resistant microorganisms. In a study, researchers found that 465 participants interviewed, 96.3 had heard the term antibiotics, and 80 had recalled having previously received antibiotics. Of the eligible 369 subjects, 32.9% of the subjects showed non-adherence, meaning that's 1/3 of the participants that did not use their antibiotics correctly.